was like, no, walking off the stairs to this class, and I was like, wait, I don't have to go this way. I was like, wait, it's
All right. Everybody ready? Meredith. Grayson. Melissa. Brina. Tana. Kristen. McKenna. Kyle. Zach. Kylie. Sarah. Ashley. Natasha. Becca. Michael. Emily. Danielle. Justice. Yes. Sorry it took me so long. Amber. And Alex. Did I miss somebody? Did I never actually write you on this list? Oh. Okay. I was wondering why I had 27 students and only 26 pages. Well, you're on here now. Yes. It's because I printed it out like before class started. So. Okay. Alright. Officially remembered everybody's names. Feel accomplished. Alright. Okay, so 2 things to start. And I announced this in lab yesterday, but for those who weren't in lab yesterday or who have lab tomorrow, there are 2 fairly minor errors on your worksheets for this chapter. The first error is that it should say DNA picograms and not pictograms. Please don't draw me nice pictures of DNA there. I want like mass measurements. So a picogram is a measurement of how much mass the DNA in the cell has. That's what I meant. And the other one is at the beginning of meiosis 2. It should be N as the ploidy and not 2N. There are some books that teach it as 4N to 2N to N. That's wrong. Don't do it that way. So it should start with N. Okay, how are those worksheets going? Good? Going-ish? I think most people have at least started them. Are they helpful? Okay. Okay, good. So what I'm thinking, and this kind of depends on how today goes, is Friday I would like to give at least half a lecture for sort of in-class working on things. That can either be problems in addition to the worksheet, it can be kind of finishing up or working through some of the questions you have on the worksheets. But I want to give a little bit of in-class time because I know people have had questions and some people have been stopping by to see me, but not everybody has that opportunity, so I'd like to be here to answer questions for anyone. So if everybody's done with the worksheet for chapter 2, then I'm happy to have people start working on the worksheet for chapter 3, which is due in a week and a half. So bring questions. Okay. Today then we are finishing up chapter 2, so we just have a couple of small things to talk about still, and then moving into chapter 3, which is our Mendelian inheritance chapter, which we'll spend a while going through. Okay, so the last little bit, one question that comes up is, how do our sister chromatids actually stay together during the process of meiosis and mitosis? And then, how do we keep them together during that first meiotic division? Right, so in meiosis 1, our sister chromatids stay together, but in mitosis, they automatically break apart. So sort of what is that mechanism? And we actually have 2 things that are working here. The thing that keeps the sister chromatids together is called cohesin. So it's a molecule. Here it's represented by these little pink circles that actually binds the sister chromatids to each other. So again, there's still physically one structure here. When we're going through anaphase, the cohesin is broken down, and so it releases those 2 sister chromatids. During meiosis, the process is similar. Okay, so our first question would be, well, how do these tetrads sort of stay lined up? And then what happens is they actually loop over each other, and then we have pieces of cohesin that bind the tetrad pairs to each other. So we're still using the same mechanism here to keep both the sister chromatids together and to keep the tetrads aligned during that first metaphase when all four of them are lined up along that metaphase plate. So what would happen in anaphase 1 of meiosis if all of this cohesin broke up?
Separate.
What would separate? Tetrads? Okay, so that's the ideal, is that the two sister chromatids separate from each other, so the tetrads separate. But what else would separate if we broke down all of that cohesin? So if we broke all of this apart. Yeah, during this first divide, what we'd actually end up with is 4 separate chromosomes. So we'd have 2 that were connected to kinetochore microtubules, and then 2 that were sort of free-floating in the cell. So we have to protect some of that cohesin during this first division so that the sister chromatids stay together, but the tetrads separate. And so we have the help of another protein, which is called shigosin, which actually protects just the cohesin at the centromeres of the sister chromatids. So these little blue dots are representing that. And so that's going to keep the sister chromatids together during that first meiotic division. The shigosin and cohesin is then broken down in that second meiotic division to let go of the sister chromatids.
You said shigosin was those dots?
Yep, these little dots. So those actually just protect the cohesin around the centromere during the first division of meiosis. And so I included, this is actually something that we didn't know for quite a while, is how this mechanism worked. In 2005 was when a paper came out that told us that we had this sort of secondary protector protein that kept those sister chromatids together during that first meiotic split. It's called the guardian spirit. It's very majestic. Okay, so the last little piece of this chapter is talking about sexual reproduction, and some of the questions on the worksheet actually get to this idea. So a couple of you have already asked me about the last 2 columns in the table for meiosis 2, because I'm asking about a zygote at that point. A zygote is going to be a fertilized egg, which means that we have a gamete from mom and a gamete from dad coming together to form what will become an embryo. So we are sexually reproducing. We are what we call heterogametic as a species, which means that we have gametes that are different between our 2 parents, or the 2 sexes that we have. And for evolutionary purposes, we define sexes by the gamete type. Okay, so a male human would have small motile gametes, which we call sperm. A female human would have large, nutrient-rich, non-motile gametes, which we call eggs. And so there are species that are isogametic, which means that everybody has the same gamete size. These tend to be our hermaphroditic species, or species that don't have dimorphic physical characteristics, like humans have a distinct female and a distinct male form. Not all species have that, so they have isogametic. They are isogametic species.
What's an example of a species?
That's isogametic?
Yeah.
It's like a lot of molds, funguses, things like that, yeah. Okay, so we're going to look at humans, since we are humans and we tend to be interested in ourselves, just quickly. The process through which we produce male gametes or sperm is called spermatogenesis. In females, it's oogenesis. And both of these processes are essentially just meiosis. One is in a male, one is in a female. There was a question on yesterday's lab or tomorrow's lab that actually asked about these two processes and how they are different or similar, right? So we actually now know how meiosis happens. When we put it into a male's body, it looks pretty much like what we would expect meiosis to look like. We start with a 2N cell, we go through a division, we now have N, so this is meiosis 1. From there, we divide again, and this time we get our 4 daughter cells, which are haploid, have a complete set of chromosomes, and then become sperm. Right? So this is meiosis as it plays out in the male body. In the female body, it's a little bit different. The end goal of having a haploid gamete is the same. There are sort of two major processes that are, or two major steps within the process that are different. So we start off the same with a 2N cell. The 2N cell divides, so here is meiosis 1, and then here is meiosis 2. I'm not sure what just happened there. Meiosis 2, okay. And that's the same for males and females, so this is also meiosis 1, this is also meiosis 2. Okay, the big difference for each of these meiotic divisions is that in a female, we take this nice big parent cell and we divide it unevenly. So we get one nice big cell, and then we get one teeny tiny cell. So what is inside of this teeny tiny cell that we call a polar body? Okay, so what is the one thing that has to split during meiosis? Chromosomes, yes. It's not just a chromosome, it's actually a complete set of chromosomes. So it's one set of those sister chromatids. All right, so we have one set of sister chromatids. Oh, these new boards are so awesome. Okay, one set of sister chromatids. I don't know what. It must be me because everybody else seems to use them just fine. All right, one set of cis chromatids in here, and then one set in here. What ends up happening is this is basically just a garbage can. Okay, so this cell dies. It's not useful to us. It's not big enough to support a developing embryo. All right, so we just use it as a garbage can for chromosomes. Right? Throw a set away. This big cell contains most of the cytoplasm from the parent, most of the other organelles, and it's nice and big and nutrient rich and can do its job. This is the cell that's going to go on and divide. Right, so it's going to go through meiosis 2. It does the same thing where when it goes through meiosis 2, we get one really big cell, and we get a second polar body, which is just, seriously guys, just another garbage can for the extra set of chromosomes. And so in a female, we end up with one viable gamete at the end, which is really big, and again has all that cytoplasm, all those organelles, and a full set of chromosomes. Whereas in males, we end up with 4 very small cells, right? The sperm cell is going to be one of the smaller cells in the human body. The oocyte or the ovum is going to be the largest cell in the human body. So they're very different in terms of size. But also, this egg floats from an ovary to a uterine tube. These sperm have to leave the male reproductive system, enter the female reproductive system, through the cervix, through the uterus, out these. So these guys are moving, and then they drop off their chromosomes, and then they just become a little wilty sac. Here we have the gamete that is actually doing the housing and the nourishing and the protecting. Right? Different purposes, different processes. So, the two major differences are going to be in the divisions themselves, right? So we always have sort of that waste cell. And then the end result is also different, where we have one gamete per division as opposed to 4. All right. And so when a sperm and an egg meet, I'm sure you know, we get a gamete, or a zygote rather, not a gamete. And this now is the parent cell for the developing embryo. Right? We start off with one cell, one set of 23 chromosomes for mom, one set of 23 chromosomes for dad, and now we have our zygote. So for those last two columns on that worksheet, you're basically starting back at mitosis. Okay, so just quickly, I think we've covered all this. The one thing that I'll point out here is we haven't talked a ton about our sex chromosomes yet. We'll get to them a little bit later. But I did mention that chromosomes 1 through 22 are homologous pairs. X and Y are not homologous chromosomes, right? They're not the same, they don't have the same genes. They do, however, still line up during meiosis and mitosis and divide so that each cell gets one copy. When gametes merge during fertilization, we either get 2 X chromosomes or an X and a Y chromosome. Right? If you get 2 X chromosomes, you're biologically female. If you get an X and a Y, you're biologically male. And so if we're looking at a female, what will her gametes look like in terms of which chromosomes end up in the gametes? Sex chromosomes. What sex chromosomes can mom contribute to her gametes?
X.
X, right? So every gamete that she has is going to have an X chromosome. What about dad? Right, so the sperm, remember he's going to make 4 of them. 2 will carry X's and 2, something about that spot. That spot's broken. Or 2 Y chromosomes.
So then it's the males that determine the sex of a fetus?
Yes. Yep. Mom always contributes an X chromosome. Dad either contributes an X or Y. So when we're talking about fetal sex, dad is going to be the determining factor. So when King Henry VIII beheaded all of his wives for having only females, that was his fault. Yeah, he should have, I don't know, tried something else. There's all sorts of like folklore, old wives' tales for how you can have one sex or the other. I hate to break it to you, but it's just random. Yeah. Okay. So in humans, sex is going to be determined by the X and Y system. Really, the big factor in humans is presence of the Y chromosome. If a person has a Y chromosome, they are going to. The Y chromosome is what contributes to what we consider maleness. Right? So in the absence of a Y chromosome, a fetus would develop female traits. Anytime a Y chromosome is present, they develop masculine traits. There are other species that use different systems, so like birds here use a ZW system. In this case, the male is what we call the homogametic sex because it has 2 of the same chromosome, whereas the female has the Z and the W. Right? So this looks different for humans, where our males are the heterogametic sex with 2 different chromosomes, and females are homogametic with the same. In birds, it's different. In insects, there's actually sort of a ratio component to it. So males have 1 X chromosome, and anytime you see this designation of a 0, that means that they just have 1 chromosome. So there's not 2 chromosomes there, it's just 1 X. So males are X, females are XX, and it has to do with the ratio of X chromosomes to the number of autosomes. Right, so if you're a male, you have 1 X chromosome, but then you have 2 copies of every other chromosome, so your ratio is 0.5. If you're female, you have 2 X chromosomes, so you would put 2 here. have 2, and then the sets of autosomes, you have 2 copies because they're diploid, so they're 2 over 2, which equals 1. Yes. I think that this means that we should be done with this. And then bees are super weird. Maybe we'll talk about bees another day. But actually, does anybody know about bees?
Annie does.
Yeah, so you talked about it, you guys talked about it. Yeah, so the way bees work is the female will lay eggs. If the eggs are unfertilized, and she's the one who determines whether they're fertilized or not, they develop into males. And so they are actually haploid, they have only 1 set of chromosomes. Females, on the other hand, are diploid, which means that they have 2 sets of chromosomes. And again, the queen will decide, like, okay, our colony needs more females because those are going to be our workers. And she actually mates once and stores all of the sperm and then like lets little bits of it out to fertilize her eggs whenever she wants. It's like a super cool system, right? But in that case, she fertilizes the eggs. Yeah, they have a parental contributor, so it's not 2 copies of her chromosomes, but the diploid is then what determines that it's a female. So the ploidy of the cell makes it male and female. All right, so all different types of sort of sex determination systems. Some of them are chromosomal, some of them are not, right? There are non-chromosomal sex determination systems, like everybody's favorites are like reptiles, yes, like turtles. No? Why is everybody staring at me like you've never heard this before? Oh no, okay. So in a lot of reptiles, it's the temperature that the eggs are incubated at that determines the sex. So in like rearing facilities, they'll have different incubators at different temperatures, and one will produce all females and one will produce all males. Yeah, so there are those would be environmental sex determining systems. All right. Cool. Learning. You guys are learning some stuff. Okay. Any questions or comments on meiosis or mitosis before we move on to Mendel, where we are going to look at meiosis and mitosis again? Okay, so I'm going to try and go kind of quickly through some of this stuff. We're going to learn things. Oh, I hate when it does transitions. I'm like, why? They always show up. Okay. All right, so before we actually get into Mendel, we are going to talk about probability and statistics a little bit. Did you learn in 100, 101, 102 your laws of inheritance? Sure. Who remembers them? Good. Not good, but. So there are 2 major laws of inheritance. We've actually just looked at both of them, so hopefully after I discuss them, they'll make a lot of sense. But one is the law of segregation, and the other is the law of independent assortment. And using this information, we can actually begin to predict what happens when individuals with certain genetic makeups reproduce. We actually do this a lot with humans. It's very cool. So we say, like, okay, you know, a lot of times before you have kids or when you get pregnant, you'll go into the doctor and you'll meet with a genetic counselor first thing, and they will say, do you want to be tested for these conditions or these genes so that we can predict sort of what the likelihood of your child having a condition would be, right? So some of the common ones would be like cystic fibrosis, like Tay-Sachs, things along those lines. And so sort of understanding how likely it is that 2 people would produce an offspring or 2 individuals would produce an offspring with a certain trait or characteristic can be important, right? So parents are interested in knowing, particularly if these genetic diseases and disorders are some of the more severe ones, like some are very easily managed, some are not. There is now a component where we're looking at personalized medicine, so we're using people's genetic information to look at how you respond to things like painkillers, right? So if you are redheaded, you actually typically respond to certain anesthetics and painkillers differently than the rest of the population. it's important to know, right, if you're going into surgery and a certain dose of something can harm you when it's fine for everybody else, that comes from your genetic information, right? And so these are just a few of the reasons why we're interested in this particular field. I know a lot of you are sort of pre-med, pre-farm, pre-something. These are just some of these sort of applied pieces. Okay. How many people have taken stats? Oh good, so this shouldn't really be new to most of you. Okay, so one of the big things that we're going to be talking about is probability, right? And we'll define probability as the chance that a certain event is going to happen, right? So we can look at this in the very sort of traditional way with a penny or a die, a pair of dice, when we're trying to figure out, well, what are the odds that we get a particular outcome? So when we're calculating probability, we take the number of times an event occurs, and we divide it by the total number of events that there are. Right? So again, to be very simplistic, if we're looking at a coin, the probability of getting heads would be 1. So that's the number of times an event would occur over the total number of events, which our events in this case would be either a heads or tails. Right? Those are your potential outcomes. And so it's 1/2 or 50%. Everybody still with me? Okay, so what would be the probability of rolling a number 3 on one die? Just one. 1/6. Right. All right, so probability can be that simple. It's not always that simple, of course. We are going to do some genetics problems coming up here in a few lectures. And one thing that we're going to start looking at is whether the results of the actual cross are different than what we would expect. Okay, so here, an example. Is everybody somewhat familiar with Punnett squares? Okay, so we will talk about Punnett squares here soon, but now Punnett squares are basically used to help us predict or determine probabilities. So let's say somebody has, actually I'm just going to ruin one of our lecture points from a different lecture right now. Everybody know what a Manx cat looks like? Okay, so Manx cats, I feel like they're cute.
So moral of the story, so you just wanted to show us what these cats look like?
No, I mean I do now, but see they got like nubby little butts. Right? So, they look like they got their tails caught indoors. What more could you want? All right, so Manx cats are a particular breed of cat. They look fairly standard except for that they have a very short tail. Okay, they're born like that. These aren't like dogs where we take their tails off. They're actually born like this. So it's a dominant trait, right? So I will use M to be the dominant, right? So if an individual has this particular allele, it means that it's going to have a short tail. Right? M would be a regular tail. Now I'm going to say that we're going to cross a parent, so a Manx mom, with a Manx dad, and both of them are what we call heterozygotes. So they have one big and one little allele, one dominant, one recessive allele. So how would we set up our Punnett square for this? Yep. Big M, little m, big M, little m. So what do these actually represent?
The alleles?
Alleles. Where would we find a cell with just one allele?
Gametes?
Yes. So when we are writing these things on the top and on the side, what we are actually putting here are the gametes that these individuals produce. Okay? So Dad is going to have, this is going to be fun, a sperm with a big M, right? Well, not A, but many, and then a sperm with little m, right? And then Mom can give her babies a big M or a little m in her eggs. Okay? So, what do we put in the middle here? What was the other big M, big M? Okay, so big M, big M. Yep. Yeah, you guys are gonna know that. There we go. Okay. Now, these are the potential offspring. Okay, so if this sperm fertilizes this egg, that baby is going to be big M, big M. Right? If this sperm fertilizes heterozygote, this sperm fertilizes heterozygote, and then here we have homozygote, recessive individual. Okay, so when we are thinking about mating these two cats together, we want to predict how many of their kittens are going to be Manx and how many are going to be normal. So how many of them are going to have long tails and how many of them are going to have short tails. We represent that as ratios. So what would be the ratio of Manx individuals to long-tailed individuals? 3 to 1, right? So if we had 4 kittens born, on average we would expect 3 of them to be Manx. We'd have 3 Manx. And 1 of them would be, we'll just call it normal for now. All right, so this is correct. This is what we would expect. You're doing great so far. Now, let's say you're actually the breeder. You breed these 2 individuals together, and you do this multiple times, and you have a nice big sample size. And what you actually find is that your ratio is closer to this. So on average, about 2/3 of the kittens are Manx, so 66% as opposed to the 75% you expected. Which brings me back to this. Here we have an expected outcome, right? We did our Punnett square correctly. We know that this is what we should expect. But reality is, this is what we got. All right, so this is an observed outcome because this is what we saw. This is what we expected to happen. We are going to use probability to determine if these are actually the same number. All right, so everybody knows that if you plant corn or peas like Mendel did and you count them, you're never going to get exactly 3 to 1, right? You get 3.1 to 1, 2.875 to 1. Right? We have a little bit of variability because it's just what happens in life. So we can test if these are the same or if there's something else going on, right? So I will tell you now that in this case there is something else going on. Right? And the thing that's actually happening is these individuals or these zygotes, this is fatal. So every big M sperm that fertilizes a big M egg undergoes spontaneous abortion and does not develop into a kitten. Right? So what we see when these kittens are born are these individuals. That doesn't mean this doesn't happen. It just, these individuals don't survive. Okay? So we'll talk about that later. But it is, yeah, sorry, did I ruin your day? Oh, kittens. Don't say that all. I should bring my cats and let you snuggle them. Yes. That would be terrible. One of my cats does not like being touched. Yeah. Okay. Okay, so, yes. This is sort of a visual representation of why we sometimes see numbers that aren't exactly what we expect. Right? Hopefully, again, I'm not very good at teaching statistics, but I will do my best. So when we are testing outcomes, we expect that most times the outcome falls within a certain range, so it falls close to average, right? So if we had 4 kittens every time, every litter had 4 kittens, we would expect some variation, right? We may get a litter that has 2 Manx and 2 regular, we may get a litter that has 4 Manx, but when we average them out, we should expect them to fall around that 3-to-1 ratio. Right? The bigger your sample size, sort of the closer to expected you'll get, right? So if you just have 1 litter, you may find 4 to 0. That's pretty far from what we would actually expect, but if you, again, have that second litter where it's 2 to 2, that now averages. So the bigger your sample size, sort of the closer you get to your expected outcome. So we can do math to tell us whether we are within that range or if we're really far outside that range. Okay, so now we're going to talk about 2 rules in probabilities. The first one is the multiplication rule. So we are going to pretend we're rolling a die. Everybody imagining rolling a die right now? Okay. Right, so when you roll a die, you will get one of these 6 outcomes. So in this case, we want to count the number of times that we get a 4. The probability would be 1 out of 6. We just talked about that. If we roll the die again and again, we want to know our probability of getting a 4. Within that roll, it's also 1 out of 6. Every time you roll the die, you get a 1 out of 6 chance of getting any particular number. But if you want to know the probability of rolling a 4 twice, then we actually have to multiply these probabilities together. Okay, so the probability of getting a 4 on the first roll multiplied by the probability of getting a 4 on the second roll is 1 out of 36. Okay, so what is your probability of having a male child at any given round of fertilization?
50%.
Yeah, okay, so 1 out of 2, right? So your probability of getting a male on the first pregnancy is 1 out of 2, and your probability of getting a male on the second pregnancy is 1 out of 2, so we would multiply those. Your odds of basically having 2 males in a row would be 1 out of 4. Okay? Easy peasy. Okay, the addition rule is next. So if we're rolling our die, okay, on average, 1 out of 6 times we get a 3, 1 out of 6 we get a 4. If we want to know within 1 roll our probability of getting a 3 or a 4, this is no longer multiplication because this is within 1 event and not across 2 events, right? The last slide for multiplication was an and. I'm sorry, it was an and. Here it's an or. So 1 or the other, 1 and the other. That's going to tell you the difference between whether you want to use multiplication or addition. So here we're adding, so we would say 1/6 plus 1/6 is 2/6, or 1/3 if we want to reduce that fraction. Okay. So here, you're going to answer this question for me now. The probability of being type A blood is 1 out of 8. Probability of being type O blood is 1 out of 2. What is the probability that you are A or O? Okay, so the first question is, are we adding or multiplying these? We should be adding, right? Because this is 1 event, and we're looking at the chance of getting 2 possible outcomes. So now, the rest of this is just, can you remember how to add fractions together? So, how do we, okay, so we got A. Okay, so how do you add 1/8 and 1/2? And I realize that this may seem ridiculous to some of you, but how do we add these
Change
things?
1/2 so it has the same denominator as well. So 4/8.
Real forceful. Is everybody with us still? All right, so your answer is A. Okay. I wish that I could tell you that it stayed this easy, but revel in it while it does. Okay. Okay, so we've actually just covered this. This is Reginald Crundle, Reginald Crundle Punnett, or something like that. Right, and so we use Punnett squares to give us answers for simple crosses. This is a very simple cross. It involves one trait. We will do dihybrid crosses, which involve two traits. How many squares do we get in dihybrid cross? 16. Yeah. I might show you a 3-trait cross, but that's, I think, 64 squares. Anything over that, I think it's a little bit alarming. Yeah, I was right. Reginald Crundle Punnett. Boom. Okay. I wish that was a lie. Okay, so we've actually already done this. So you can kind of fill this in on your own time if you want. We've already done our Punnett square how-to. Example. We're going to make a lot of Punnett squares, so don't be alarmed that I just brushed past that. Okay, so now we're going to practice the multiplication rule. In this situation, we are going to be looking at a disease called congenital analgesia, which is those people that don't feel pain. Yeah, so everybody's seen like that House episode with the kid that feels no pain, breaks things. It's not very common, but it does exist. So people don't have pain receptors, or their pain receptors do not transmit the right signals to the brain, so they don't know that they're in pain. It's actually very dangerous. It's not good to not feel pain. You feel pain for a reason. So it is a recessive trait. The normal allele is big P. The recessive allele that would give us this trait would be a little p. So my question is, you have two heterozygous individuals. They want to start a family. What is the probability that their first 3 children will all have congenital analgesia? All right, so both parents are carriers, which means, so what would the genotypes for these individuals look like? Big P, little p, right? So they have one allele that's a big P, one allele that's a little p. You can talk to your neighbors if you're not sure what's going on. Or if you want to verify. Give you a second to work it out. It really helps sometimes if I look at my notes. I don't know if my math is okay. So everybody got it or got something or completely stumped? One of those 3, everybody is in there somewhere, right? Okay, so the first thing that you want to calculate are probabilities for the individual offspring. And in this case, we're really mostly concerned with offspring that are potentially going to have this particular condition. So what is the probability that one of their offspring has, I have a meeting or something coming up. Okay, so what is the probability that one offspring has this condition? 1 out of 4, right? So 1 out of 4 for an offspring. So then how do we go about figuring out if multiple offspring are going to have this particular trait? So the first 3 offspring, what are the odds that they all suffer? Power of 3. Okay, so I heard this. You know what's funny is I'm actually probably one of the better versed computer people in this. What am I doing? Alright, so I heard this. Yeah, right, which is the same thing as. Okay, so there's a 1 out of 4 chance that the first one has it, 1 out of 4 the second, 1 out of 4 the third. So what is the probability that all 3 of them together have it? Yeah, 1 out of 64. We can convert these to percentages because for most people, a percent means more, right? If I say you have a 1% chance of, I don't know, rolling your ankle today, that sounds a lot different from you have 0.01 chance, 1 out of 100. Right? So when we convert this to a percentage, nope, 1.6. 1.6%. Right? Okay. Great. So now, calculate the probability that you have 3 offspring. The first one, 2 of them are affected and 1 of them is not. So you have 2 offspring that are, what did I say?
2 affected, 1
Okay, that's not what I wrote, so I'm going to go with what I said. 2 affected, so 2 that have the condition and 1 that does not. How would you calculate that? All right, somebody say some numbers. 1/4. Right, so this is an affected individual times an affected individual times a frozen computer. Right, so you would do 1/4 times 1/4 times 3/4. And I don't have the math on that, but it is 3 out of 64. And so these are the types of problems that some people want to know, right? You might want to know if you have a 25% chance of having a kid with a potentially life-threatening condition, right? Okay. So I have 1 minute, and so I'm very quickly going to go through the theories of inheritance because they're kind of cool, and I have a minute. When we're talking about Mendel, we're going to be talking about the actual theories of inheritance, but before Mendel, we had lots of kind of very interesting theories about how people inherited conditions from their parents, because we didn't know what gametes were. You can't see a sperm cell like with your naked eye, right? So one idea was called the theory of pangenesis, which was that basically cells from all the different parts of your parent's body traveled from that location to their reproductive system, and then out the reproductive system and merged into, it's not like totally, it's pretty bad, but my favorite is preformation, which was this idea that a tiny miniature living person was in a sperm cell. So each sperm cell had a little teeny tiny baby in it, and then it just had to go into the female, and then it grew there. I love it. This little guy. It looks like the one where you see that in like cartoons and stuff still. And then the last one was probably the one that maybe some people know of, blended inheritance. So that would mean that you're essentially the exact intermediate of your parents, right? So if your dad is 6 foot tall and your mom is 5'6", you would be 5'8", because you'd be exactly the center, right? We know that that's not true, but we had this sort of idea that it was somewhere in that sort of range. All right, so we will start on Friday with Mendel and his keys. Again, if you have questions or anything you want to know about those worksheets, I'll give some time on Friday to work on them. Yeah. Sure.
I was gonna say, I didn't like look at the Dropbox.
I'm not going to grade a lot of
No, like Friday after, like we don't even be talking about it on Friday.
Have a good day. So you mean like this number? Yeah, so this is individual. I know. Are you too? Yeah, no, that's fine.
Oh, see, I just so like when everything like duplicates, so like if you like how like it goes from 46 to 92. So when it's yeah, but so are you saying that the top would stay 6 and then the bottom one goes to 12?
Yes.
Okay.
Yeah, because the top is chromosome.
Yeah.
So whether it's 1 or
Yeah.
here it's still 1.
Yeah, I just want to make sure that I was thinking it right. With you saying that, now like
DNA
that
molecules
confuses me.
is another way to say chromosome.
Yeah.
Yes.
I just need to print out that. I just believe something. Is there somebody there? I can listen. Yeah. I'm so lazy.
Oh no, I'm sorry.
I forgot my notes.
So it's the picogram is actually you should use
I gotta tell him about my hot sauce.
Oh my god. Sure, take a seat.
Thanks. So I had this hot sauce. I put down my story for my try.
I really hate hot sauce. Okay.
Don't eat it, okay? So I had chicken, right? And I just put a little bit of this on the plate, and I took like a thumbnail size swipe of sauce, okay? And I like looked at it and I was like, man, this stuff like smells hot and it looks evil. And I took a bite of it and like instantly like it was like almost like
Death nectar.
and then yeah.
You didn't think that was spicy?
No, I signed waivers for hot sauces and they weren't even that.
I would have
Like,
not. I think
okay.
garlic parmesan.
So then my eyes like started watering, like my face, like I had just snot running down my nose, and like I went straight to the fridge and just got milk. I went through 3/4 a gallon of milk. It's like swishing it in and out around my mouth, and then I went through 3/4 a gallon. Dude, it was bad. It was so hot.
Sorry, we do
And then the worst part was this morning.
Oh yeah, you gotta listen to
Okay,
this
so I
part.
woke up this morning, I had a pee super badly.
Oh no.
So I walked and I started pissing. Dude, it burned so
Do you have
bad.
a UTI?
Dude, I don't think so. I was like, dude, it burned so bad. I had
Can you like get out of my chair? I was like, you're just like, can you just...
I could for so long.
It's
Like
great.
it burned. Like I can't say I had pee burn. Is that normal?
Yeah, prediction.
Dr. Brian, is this still recording?
Yeah.
That's fine.
I have a great story.
I don't really care.
I totally just to the last 5 minutes of that.
Is it-
Don't, you might want to delete the
I got a
last
question.
like 5
You
minutes.
probably know the answer. So, let's say a friend of mine or myself just ate some really hot sauce and my pee burned this morning. Is that normal?
No. I have never heard of hot sauce.
It's called Death Nectar.
It has ghost chilies in it. I wouldn't recommend it.
I would expect the burning to be not from urine.
Yeah, I really no. It burns like everywhere.
Well, that would
I
make
can't believe you
Yeah.
just asked her.
Like
For a friend. Did you watch
You went to the bathroom?
yeah. No, I went to the bathroom in the morning. Like I washed my hands and everything when I was